welcome to Osteobites. Happy Thursday. My name is Christina Iptoma, and I am mom to Osteo Angel Dylan and Director of Scientific Programs at MIB Agents. And today on Osteobites, we are talking with Dr. Michael Kinnaman about how osteosarcoma evolves from diagnosis to relapse. Thanks so much, Dr. Kinnaman, for joining us on Osteobites today. We are thrilled to have you. And we're also very happy to be joined by our panelist, Mia, who's a fierce osteo warrior and vice president of our MIB Junior Advisory Board. A little bit more about our guest today. Dr. Kinnaman is a member of the Pediatric Sarcoma Service at Memorial Sloan Kettering Cancer Center in New York, where he specializes in taking care of children, adolescents, and young adults diagnosed with pediatric sarcomas. He's a member of the Iacobuzio-Donahue Lab Group, with research efforts focused on describing and detailing the clonal evolution of cancer cells in pediatric sarcomas. Dr. Kinnaman has been the recipient of a number of Young Investigator Awards to fund his research, with support from Hyundai Hope on Wheels, the Raleigh Foundation, ASCO Conquer Cancer Foundation, and the QuadW Foundation. And he's also been awarded a National Institutes of Health MSKCC K-12 Paul Calabresi Career Development Award for Clinical Oncology. Welcome, Dr. Kinnaman, and welcome to everyone joining us today. Please feel free to add your questions for Dr. Kinnaman to the Q&A as we go through the slides today. And before we get started, just a few announcements. If you haven't already, please subscribe to our monthly newsletter called Connective Issue. It includes info of interest to our osteosarcoma community, including recent news and papers on osteo, open clinical trials, even job posting in labs that are working on osteo, info about MIB events and programs, and also the upcoming Osteobites schedule. Our second issue just went out last week, and I'll put a link in the chat if you're not already getting it. I'll put a link to the newsletter, and then you can just also go to our site and hit the Join Our Community button to subscribe and just get it in your inbox every month. And then we also have our virtual tumor review board for osteosarcoma coming up on November 9th. If you are a clinician or researcher with an interest in osteosarcoma and would like to be added to the calendar invite, or you are interested in submitting a case, just fill out the form on our site, and I'll put the link in the chat as well. And for anyone in the Houston area, we are going to be heading your way next month. Join us on November 30th from 5 to 7 p.m. for an MIB Mingle at the Blossom Hotel, which is right across the street from MD Anderson. Everyone in the osteosarcoma community is invited, and we'll be sending out an invitation, but I'll just put an email address in the chat if you have any questions or wanted more details. And we'd like to thank the sponsor of this episode, the Osteosarcoma Institute, OSI, which is a nonprofit organization led by osteosarcoma experts from top U.S. cancer centers who together are concentrating on the cure for osteosarcoma. The mission of the OSI is to dramatically increase treatment options and survival rates in osteosarcoma patients through identifying and funding the most promising and breakthrough osteosarcoma clinical trials and science. And in addition to advancing research, OSI also provides a free resource called OSI Connect for osteosarcoma patients. Their osteosarcoma experts can discuss available treatments, possible side effects, and provide helpful advice for getting the most out of your visits with your treating physician. This resource is available in English and Spanish and aims to help patients and families find answers to their questions. So enough from me. I'm going to hand it over to Mia to introduce herself.
Hi everyone, my name is Mia Sandino. I'm 23. I live in LA with my mom and our 4 cats and 2 dogs. We have a zoo. I am currently in between treatments and in between school. So I like to refer to myself currently as a non-practicing intellectual and as the vice president of MIB's Junior Advisory Board. It is a pleasure to be here today. And it is an honor to introduce Dr. Michael Kinnaman. I can't wait to see your slides. I've already perused them a little bit, and I can't wait for you to share them with everyone else.
Wow. Thank you, Mia and Christina, for those very, very kind words and introduction. Very excited to be here. The big fan of the MIB Foundation. I point many of my patients to the website. The resources are incredible, and thank you to you both for everything you do for the community. All right, so as mentioned before, my name is Michael Kinnaman. I am part of the pediatric sarcoma team here at Memorial Sloan Kettering, and I am going to talk to you all today about how osteosarcoma evolves from diagnosis to relapse. So kind of to help frame my research in osteosarcoma, I think it's important to kind of review some concepts from evolutionary biology. And these are things I hope many of you are familiar with. Maybe you learned about them during high school biology, maybe saw a documentary. And if you're sitting there thinking to yourself, that guy on the left there, he looks familiar. Is that Charles Darwin? You would be correct. That is Charles Darwin. He is the kind of the godfather of modern evolutionary biology and how we kind of think about evolution in nature. And I think he's best known for this concept of survival of the fittest, right? That's a term that's often thrown around in many different scenarios and settings, right? So we hear it in sports. Like I often think about it, in context of New York City playgrounds. Whenever my daughter goes to play, it's all about survival of the fittest. So Charles Darwin, how he kind of got to this was very smart man. He would head down to these tropical islands down, and they're called the Galapagos Islands. I'm sure many of you have heard of the Galapagos Islands, and they're off the coast of Ecuador. He would head down there with his notebook, and he was a master observer. He just would go down and he would observe the animals down there, their environments, and he would hop from island to island and kind of write down what he saw, write down the characteristics of the animals. And he began to develop some observations about what he was seeing. And one of the observations he made, something quite simple that we would kind of not put too much thought into, but he noticed that traits were passed down from a parent to their children, from to their offspring, right? So for example, if you're a bird and you had a long beak, right, your children would have long beaks, right? And at the time, he didn't know about DNA. It hadn't really been discovered or described. And so he didn't know how it was passed along, but he just knew if your parents had long beaks, then the children would have long beaks. He also noticed that, take a species of birds on the island, right, that they are not, they weren't all the same. Some had different characteristics. And what would happen, or what he noticed, was that as time would go on, and maybe the resources for those birds or the food that they were trying to obtain became more limited and the number of birds increased, right? Then those birds that had, let's say, the beaks, for example, which allowed them best to obtain their food source, maybe that long beak allowed them to get to hard to reach seeds or grass or whatever it may be. The birds with the characteristic that allowed them to obtain their food and their from the environment that they were in were the ones that would survive, and the ones who didn't have those features would become extinct, right? And that's this concept of survival of the fittest. And we now know that what causes this variation amongst species, the different traits, the different characteristics amongst us and amongst animals are mutations, right? We're all born with DNA. That's our genetic code, and it has all the coding for all of our features and characteristics, right? And over time, randomly, mutations get introduced, and sometimes a mutation is introduced and there's really no consequence. It doesn't really give us a survival advantage, or doesn't give us any disadvantages either, right? And those are called neutral mutations. I think of that like eye color, at least for now, right? If you have brown eyes, you're no more likely to survive in this world than if you have blue eyes, and vice versa. Sometimes a mutation may give you a feature which gives you a survival advantage, right? So again, like a beak that would allow a bird to obtain their food better, that would be advantageous. Or deleterious, where maybe you develop a character mutation leads to you to develop a characteristic that makes it more difficult to obtain food, and you would you would then not be able to survive in that environment. And these mutations have to be taken into the context of whatever environment you're in, right? When Darwin was going from island to island to island, the unique thing about the Galapagos Island was that the different islands had different environments, different resources available. And so the animals there would all adapt to the environment, and the mutations that caused the different traits had to be viewed in the context of the environment that they were in. And so this kind of process of, mutations creating variations in the population, the resources becoming limited in an environment for these species or for these animals, and whatever animals had the characteristics which allowed them to survive, those characteristics would be selected for. That's natural selection. Those animals would be able to survive, produce offspring, and eventually those who didn't have those features would become extinct, and the ones with the features were selected for, natural selection. And that's really where Darwin noticed, by observing finches, which are birds, and the different beaks and how they adapted to their environments, he observed this process kind of unfold. And there's a lot of fascinating examples of this throughout history. If you look back in time to the Industrial Revolution, this was 1800s. There was no regulations on environmental laws, right? And before the Industrial Revolution in the United Kingdom, most of these moths were white in color because it allowed them to better blend into their background and hide from their predators. The Industrial Revolution happens, and the few moths that were around that had black color as a result of a mutation, right? They got selected for because they could now hide in that background of black soot that was all on the buildings during the Industrial Revolution, right? So they had a characteristic, a mutation, which gave them that black color, and the environment changed, and that gave them a survival advantage. So why am I talking about all this? How does it relate to cancer, right? Well, we see the same sort of process unfold in cancer, right? When a tumor, when you're diagnosed with cancer, there are millions of cancer cells, right? And they all have genomic features or mutations that they share that led to the development of the cancer, right? They all share those mutations. But over time, each cancer cell may start to develop its own unique set of mutations as it kind of grows and divides. And the same thing happens as we talked about before with mutations. Some mutations are neutral, meaning they don't give an advantage or disadvantage to the cancer cell. Some are deleterious, meaning they lead to the cancer cell dying off. And some give the cancer cell a survival advantage. And what we see over time is that with treatment, the cancer cells that have that survival advantage get selected for and begin to grow out, right? And this is thought of as treatment is thought of as a bottleneck effect. And what I mean by that is that before treatment, you start with a kind of a diverse population of different cells with different characteristics. We give treatment, and for osteosarcoma, we give chemotherapy, right? And that creates a major reduction in the size of the population of tumor cells, right? And as a result, we're left with very few tumor cells, and our hope at this point is that the few that are left do not have the necessary characteristics or have not made the adaptations or don't possess a mutation to allow them to survive and come back, right? But and we hope that they become extinct. But sometimes they survive and then they come back, and that's what happens at a relapse, right? Those cancer cells from the beginning survive. They developed a characteristic which gave them a survival advantage, and that's what leads to relapse. And we can see examples of this in nature too, right? Like the most obvious one that comes to mind is the asteroid hitting Earth, leading to the extinction of the dinosaurs. And that's considered a bottleneck event, right? It was a major reduction in the population of dinosaurs, and the dinosaurs did not adapt and became extinct. And with cancer, right? The bottleneck event is treatment, and you have one of two results, one or two pathways as a result of this. Either our hope is that the cancer cells become extinct, but unfortunately, sometimes there are cancer cells that survive and they begin to recover, and they all possess, the ones that remain, those features that allowed it to survive. And that kind of gets us to where, the rationale or the reason or why what my research kind of focuses on, right, is like what changed, or what is the genomic characteristic for patients who have their osteosarcoma relapse? Like, what is it about those tumor cells that gives them that survival advantage, right? So if we can figure that out, then maybe we can figure out how to target those cells that have that survival advantage and drive that tumor cell population into extinction at the time of, your first round of treatment, right? So transitioning now to my current work and my current research, the kind of the motivation behind it, we can kind of go back to 2016. I was a fellow, first year fellow at Memorial Sloan Kettering, and my very first patient that I picked up as a continuity patient, right? So when you're a fellow, you kind of pick up patients along the way that you kind of become their go-to doctor for. And for me, this was a young boy with osteosarcoma. He was around 11 when he was diagnosed, and he had localized osteosarcoma, and he went through MAP chemotherapy, did well, and about 18 months from when he was first diagnosed, on a surveillance scan, his cancer came back, and he was the first patient I took care of that relapsed. It was devastating. I was close with the family, and although it was difficult for me as a first year fellow to kind of process, I also found myself motivated to begin to understand why this happened, what changed. And so when you're in fellowship, your first year you're in the clinic a lot, but your second 2 years they design it in a way where you're supposed to start pursuing a research question or a research passion. So for me, this is kind of what led me to pursue doing research in osteosarcoma. And so kind of untraditionally, I joined a lab of Christine Iacobuzio-Donahue, who is a world leader in clonal evolution, or how tumors change over time research. And although her main focus is in pancreatic cancer, right? Just like the principles that kind of Darwin first described can be applied to how we think about cancer, the same principles that and tools and techniques that they use to study pancreatic cancer can be applied broadly across many different kind of cancers. And I really thought osteosarcoma would be a good model to study clonal evolution, or how the tumor changes over time, because one of the most important and somewhat unique characteristics or features of the management of osteosarcoma, right, is if your osteosarcoma returns, say if it's in the lung, we go in and we take it out. And if it comes back again after that, odds are we're going to go in and we're going to take it out. And as a result, we have tumor samples from multiple time points in a patient's treatment or throughout a patient's disease course, right? So we will have the diagnostic sample, we'll have a resection when you get your primary resection done after 10 weeks of chemotherapy. And then if your tumor comes back, then we get tumor samples from multiple time points. And once we have tumor samples from all those different time points, then we can kind of do ask a simple question of, what are the genomic changes of the kind of relapse sample compared to when you were first diagnosed? And so to begin to answer this question, I went digging in the crates at Memorial Sloan Kettering, went into our biobank, and I was looking for patients where we had samples from multiple time points. And I really was hoping to get patients where we had a pre-treatment sample, an on-therapy resection sample, and at least either one metastatic sample or one relapse sample. And, I know many of the patients and their families listening in, when you consent to, allowing research to be done on your tumor, this is the kind of work we can do because, all of these patients here, gave us permission to use their samples, and I wouldn't have been able to do any of this work without the kind of the patients allowing this. And so thank you to all of you who've signed and allowed us to begin to study some of these questions. And here you can see my cohort of patients. It's 8 patients, and the samples and the time points, the number of samples at each time point that I have for each patient. So the darker the blue means we collected multiple samples at a particular time point, the most being 5 we did kind of multiple samples from a primary resection. Here for this patient, we had multiple metastatic sites, a number of 4 at this time point. And then some of them have 1, 2, 3, up to 4 relapse samples that we were able to obtain for further analysis. And as far as like the disease course for each patient, this kind of shows you the amount of time from, in blue is diagnosis to their primary resection. If they had metastatic disease, then the orange highlights the time from primary resection to the time we took out their metastatic disease, and then the interval between either whether they were disease-free or they had a relapse. And you can see that some patients had this very like slow relapsing remitting course where they'd get treated, we do the surgery, and then maybe a year or 2 later it would come back. And this kind of happened many times over the courses, over 5 years. And some of these patients had really aggressive disease where, the time from diagnosis to unfortunately the patient's death was less than 2 years. So we had a wide variety of different types of osteosarcoma that we see and that we manage. So we thought this was a good representation of osteosarcoma, disease courses in general.
Mm-hmm. Hey, Dr. Kinnaman, you might be getting to this, but in that slide, were there different interventions like at each relapse, like additional treatment that would create another bottleneck?
Yes. I have 2 slides from now.
I will wait.
They'll have the treatment. Yes, no, but good question. And so our analysis was focused on studying the major variant classes. So taking a couple big steps back, some of you may be familiar with genomic sequencing. I know many of your providers will often consent to you to getting genomic profiling done on your tumor, right? And what that means is, we're all born with our genetic code, as I was speaking to you all about before. But the tumors, they have kind of a unique genetic code, right? Because they have to develop osteosarcoma, you need mutations in your genetic code, which kind of leads to this uncontrolled kind of cell growth and division of unorganized bone cells, right? Like that's a very kind of simplistic description of what leads to osteosarcoma. There's a mutation happens typically at random that gives these bone cells, which haven't kind of fully matured, they start to grow and divide before they're fully mature, and in an uncontrolled way, right? And probably from 2014 on, a lot of studies were done trying to profile what were these mutations in osteosarcoma that could explain or could have led to that patient developing osteosarcoma. And there are different ways the DNA can change. The way on what's described on the left is called a single nucleotide variant, or sometimes it's referred to as a point mutation. And our genetic code is made up of different arrangements of 4 different base pairs, A, C, T, and G. So that's all our genomic or our DNA is. It's just long stretches of bases of A, C, T, and G, right? And in some instances, like the T becomes an A, and that leads to a mutation, which may give, a survival advantage, right? So those are called point mutations. T becomes an A, and they're kind of one base change in the genomic code. Now, if you may remember from high school biology, we're born with 46 chromosomes, half from mom, half from dad. And this is where the chromosomes are long stretches of the genomic code. And sometimes long stretches of the genomic code can get deleted. And sometimes they can get duplicated, meaning, say take chromosome 8, maybe part of chromosome 8 here, an event happens where now you have two of these sections of chromosome 8. Maybe it gets amplified again and you have 4, 8, 16. And this happens in osteosarcoma. This is actually a big genetic change that happens in osteosarcoma, where you get multiple copies of the same chromosome, and it just leads to kind of more resistance and uncontrolled growth and division, right? And sometimes what we actually think happens in osteosarcoma early on is, for lack of a better term, a bomb goes off in the genome and it gets stitched back together in ways that it shouldn't. So say this is chromosome 17. You have mom's 17, dad's 17, right? But the bomb goes off, and as your body, as you try to repair it, you attach a little bit of chromosome 16 here, and it creates these kind of unique either genomic fusions, insertions, deletions, and it can just make the genome chaotic. And that's how people often describe the osteosarcoma genome. It's just chaotic because of these, because of this, for lack of a better way of describing, like the bomb going off, rearranging all of the chromosomes in the tumor cells, and it leads to the creation of the amplifications, the chromosomal rearrangements, and so forth. So we did whole genome sequencing, meaning there are different types of sequencing that you can do. You can do very targeted, where we're just looking at specific mutations that we know can cause cancer. At Memorial Sloan Kettering, this is called MSK Impact. Foundation Medicine's a popular one. Those are targeted panels. You can do something called whole exome sequencing, which does sequencing of all the coding regions. So your genome is divided up into coding and non-coding. Most of your genome is non-coding, meaning it doesn't code for a protein. And whole exome is doing all the coding regions, and whole genome is doing all the coding and non-coding regions. And the advantage of whole genome is that it allows us to really describe all of the copy number variations and the chromosomal rearrangements. And because these are big recurrent events in osteosarcoma.
Dr. Kinnaman, I have a quick question. When you refer to kind of like the bomb going off, would that be, for example, like a stressful incident that happens in the area? Like, for example, about a year before I was diagnosed, I had sprained my MCL in my right knee, and I ended up with a 2.5-inch tumor in my right proximal tibia about a year later. So would something like stress exacerbate that kind of bomb going off? Or would you say it's just something that's more random and chaotic than that?
I think the latter. I would say that we have not traced osteosarcoma to back to stressful situations like you described. What we think happens is you get a mutation, because most osteosarcomas will have some sort of mutation in a very important gene called TP53, or which is if it's working, if TP53 is working, it's our best, or one of our best tumor suppressor genes. But once that gets altered, it leads to proliferation of cancer cells, and that's probably an early event. And once you have that P53, then it kind of is a setup for this event to occur. But we're still just like beginning to learn about it's called chromothripsis, but like it's interesting that we think it helps us begin to understand like how we get from a normal kind of bone cell to osteosarcoma, and we're just beginning to describe that transition. But I don't think we can relate it back to stressful events, as you were asking.
Gotcha, thank you.
And Dr. Kinnaman, I had a question too. I appreciate the slide to kind of, especially for me and other lay people in the audience, just kind of sorting out the different types of mutations. But just to make sure I understand, so there's the single nucleotide variant where it's just like rearranging one of those letters. There's the copy number variants, which are deletions, duplications. And does that, when we hear terms like, amplification, gains and losses, where do those fit in this?
Those would be copy number variants.
Okay, so like more copies or fewer copies.
Exactly. So like a lot of these terms are interchangeable. Like duplications can be referred to as a duplication, a gain, or an amplification. An amplification is typically a higher number of duplications than a gain would be. There's certain thresholds that we use to determine that. But I can imagine it gets very confusing as a parent or a patient trying to decipher through, especially when there's 5 different ways of calling something the same thing.
Yeah, and the rearrangements then are the fusions or translocations that. Okay.
Exactly. Yep. Another fusion, translocation, another genomic event that has 5 different names.
Right. Okay. Thank you.
No problem. So this is called an oncoprint here, and these are for all of the samples. So there was 37 samples total. And from going left to right here is each patient, and their samples are tied together. So for example, this patient is OSCE 1, there's 6 samples, and these are their characteristics, right? And the take-home message I want to kind of convey here is that a couple things. So first, 4 patients had localized disease and 4 patients had metastatic disease at diagnosis. So again, a good mix of the different types of osteosarcoma we see. And what's listed here are the single nucleotide variants that we were just talking about, and the structural variants that we were just talking about, or the chromosomal rearrangements for each patient. And what I say up here is typical for osteosarcoma, meaning P53 in all but 1 patient was mutated. And this is the kind of number we're quoting these days is probably upwards of 90% or more of patients have some sort of mutation or disruption in P53, which is frustrating because it's one of the genes that we can't target with the drug yet. We're still trying to figure this one out. And then a number of other mutations that are common in osteosarcoma, such as RB1 is another tumor suppressor gene that is often mutated in osteosarcoma, ATRX. Again, another one that's commonly recurrent. So these are typical mutations that we see in osteosarcoma. And what we noticed was that they were almost always present from diagnosis and to relapse. And there were no new point mutations or single nucleotide variants or structural variants that we detected in relapse samples, which we could point to and say, hey, maybe that's what's driving the relapse. So like the mutations in, just to go back a second, in the SNVs and the chromosomal rearrangements, those were there from the beginning and didn't change with time. That will become important in a second. And then they're getting back to Christina's question before, these are all the different treatments they receive. So every patient started with MAP chemotherapy. Some were switched to ifosfamide etoposide if they had disease progression while getting MAP chemotherapy. Some received that at relapse. For some patients who relapsed, they had a period, they just did observation after we did their resection. And then these are all kind of second, third, fourth line therapies that have been either trialed or used or used typically for recurrent osteosarcoma. So again, like a representative kind of group of osteosarcoma patients. And so
before we transition, we have a great question that came in from the audience.
Sure.
So how does somatic RB1 mutation affect the relapses? And what is the relationship between somatic RB1 mutation and relapse? And do you know of any clinical trials to cure somatic RB1 mutations?
So somatic RB1 mutations are common. They're the second most common mutation in osteosarcoma, and they can happen in different forms. They can be single nucleotide variants, they can be structural variants, or deletions. And like TP53, because it's a tumor suppressor gene, the problem with tumor suppressor genes are that they need to work to when they're working, that's a good thing, and they're able to suppress cancer or cancer proliferation. When they don't work and when they're mutated, there's nothing, we have not been able to figure out essentially how to turn these genes or these tumor suppressor genes back on. But it's like a very active area of research. But it's kind of been, if we can figure out that, then we're going to be onto something. But tumor suppressor genes, as opposed to a different classification of genetic mutations called oncogenes, which with an oncogene, those mutations essentially they make a certain path, they overactivate a certain pathway, which leads to more growth and division. So they're kind of ratcheted up, and they're overproducing whatever protein or signal that they code for. And when you have something that you're overproducing, the next question is, well, can we block whatever that signal is trying to target? So that's where like targeted therapy comes in. But targeted therapy really only works at this kind of moment in time on genes that are activating something. If you lose the function of a tumor suppressor gene, we can't turn that function back on. We haven't figured that out yet. So unfortunately, that's my long-winded way of saying we at the moment have nothing for patients with RB1 mutations. So keeping all of that in mind, right? So the take-home message was the SNVs don't change. The structural variants really don't change from diagnosis to relapse. So the next question was, well, do the copy number alterations change, right? And this is a plot of my cohort here of the 8 patients and their copy number alterations. And remember, we have 23 chromosomes, or 46 chromosomes, 2 from each parent. These are the chromosomes listed here. And the number of samples that with gains versus deletions are kind of plotted across the different chromosomes. And the whole point of the slide is just to show you that when compared to another big osteosarcoma study, the peaks where we see recurrent kind of gains and amplifications, and the valleys where we see recurrent deletions are very similar to previous studies. And again, just trying to highlight that our cohort is kind of representative of what we typically see in osteosarcoma. So now this is kind of where, for me, the kind of fun and exciting part came in, right? Because I knew copy number alterations were going to be an important kind of genomic alteration to kind of describe. And so as a plug to any of you who are kind of trying to figure out what you want to do in life, if you're really into like computers and coding, and you're also have an interest in science and cancer, there's a beautiful field called bioinformatics, which kind of links the two. And I've had the pleasure of working with a lot of very smart people in the world of bioinformatics, and they create these tools which allow us to ask questions of the genomic sequencing data, right? So right around the time we were doing all of this genomic sequencing for these patients, I wanted to know a very simple kind of question, right? Or a tool came along that helped me answer a question that I had, right? And if you think of it this way, getting kind of back to some of the stuff we talked about before, if I have 100 tumor cells, let's say, can I figure out using a tool or computational tool how many of those tumor cells have copy number profile A, let's say, which maybe doesn't really have any interesting or unique features, and copy number B, which for argument's sake, let's say has a MYC amplification, which is something you may have heard about on this call previously, is something that's talked about a lot in osteosarcoma, right? So out of my 100 tumor cells, how many have, a copy number profile that doesn't have that MYC amplification, and how many do? And so this is postdoc, or this scientist, Simone Zaccaria, created a tool that could do exactly that. And what's beautiful about this tool is that it takes all of the samples from one patient, and it says how many different copy number profiles, how many different, and these are, we would call these clones. A clone is a tumor cell that has or a group of tumor cells that share the same genetic features. How many different copy number profiles are present in this patient's tumor, and in what proportion over time are the different copy number profiles? So like, say I have at diagnosis, 99 out of 100 99 out of 100 tumor cells have copy number profile A, and only 1 out of the 100 have copy number profile B. Well, this tool can tell me when we go in and get that next sample, well, now actually 99 out of the 100 tumor cells have copy number profile B with that MYC amplification, and only 1 has copy number profile A, meaning that maybe that MYC amplification was giving that tumor cell a survival advantage, and it was selected for. And I'll show you what I mean by this. So this is one of the first cases. This is OSCE 1, and we had 4 relapse specimens, as well as a biopsy and a primary resection. And what I'm showing you here was that the tool said, okay, we can identify 2 different copy number clones, meaning, getting back, at diagnosis, this patient had 99 out of 100 tumor cells had this copy number profile. And then with treatment, we began to see copy number this copy number profile, which was only present in a very small percentage of the tumor cells at diagnosis. It begins to emerge and then kind of dominates through these next couple of relapses. And I think the next natural question becomes, well, what is it about clone 2? What is it about this copy number profile is there anything that gives it a survival advantage? Getting back to some of the stuff we were talking about before. And so we then kind of look at the different copy number profiles for each clone. And this is going to be probably a confusing figure for you to understand, but essentially the teal here, and these are the alterations, represents this clone 2, has a couple of very unique amplifications in genes that we know play a role in osteosarcoma. So this patient had a CCNE1 amplification, a MYC amplification, that were in this clone 2 that were not in this clone 1. And so that, you begin to kind of think to yourself, is this just a unique feature of this patient, or is this something we see across many patients, right? Here is OSCE 6. We had a biopsy, a resection, and a relapse. And again, getting back to that example, 99 out of 100, let's say, had these genomic features of clone 1 at diagnosis, and only 1 out of 100 had the features of clone 2. We give our 10 weeks of chemotherapy before you have your primary resection, and this clone 2 begins to emerge and dominate. And then at the relapse, it's again this clone 2 that is a majority of the tumor cells. So again, what's it about clone 2? Clone 2 has this IGF-1R amplification that this first clone did not possess, as well as again a MYC amplification. So we're starting to see, right, just to review, the single nucleotide variants don't really change with time. The structural variants don't really change with time. But what we see here is that there's a small population of tumor cells at diagnosis that contain these really aggressive copy number mutations. And even after 10 weeks of MAP chemotherapy, they're selected for and they're emerging, and then they're dominating. So that's kind of like, if you're asking what's changing, it looks like, you know, in this small population or small number of patients, it's the copy number alterations that are changing. Then we had the opportunity to ask the question, well, when we go in and do our primary resection, right after that 10 weeks of chemotherapy, is there any differences in the genomic features depending on where we sample from, right? So when your sample goes for genomic sequencing, they just choose one of these sections here that has, you know, the most viable tumor, the most tumor that they think will be acceptable for sequencing, and they run it. But, you know, that's all well and good if all of these different sites have the same genomic features. And so what we show here is that for the most part, they do. And that kind of makes sense, right? Because at this time point, the primary resection, 10 weeks of chemotherapy, the bottleneck has happened. And what's left are, there's not much diversity left. And these, you know, kind of characterize what we think were going to be the resistant cells. But you do see here, you know, if you had just sampled M here, you would have missed this clone 3, which then emerged and dominated at relapse. So while they are mostly, you know, similar, there can be instances where you might miss something depending on where you pull that sample from. And again, the features of this clone 3 were, you know, MYC amplification that was not as high or seen in the other clones. And this MYC amplification, this MYC gain, again, is something you've probably heard about before. It was often seen in what I call the chemo-resistant clone, the one that kind of emerged or dominated at relapse. And it was seen in multiple patients as being a genomic characteristic of the resistant clone, in addition to other alterations. But that was probably the most kind of recurrent theme that we saw. And we know that, you know, survival and outcomes for patients with this alteration they have lower long-term survival. Patients we're beginning, this is unpublished data, but data that is beginning to take form, that it may be what we would call a poor prognostic, meaning patients with this alteration may not have as a high chance of surviving long-term as patients who do not have that alteration. And so what does this all mean? Like, let's think about this practically for a second, right? And trying to put everything back into the context of what we discussed before. When we give chemotherapy, right? MAP chemotherapy, you're all familiar with. Our hope is that it delivers what we call a successful first strike. And we create the bottleneck, and we drive the tumor cells into extinction. Unfortunately, what happens with patients who relapse is that we create the bottleneck, but then we have the survival of the fittest problem, where we are unable to make that tumor population extinct, and the fit cells then proliferate and come back at relapse. So how do we prevent this from happening, right? And one of the strategies that can be used is, well, we have our first strike. We know MAP chemotherapy works well for a lot of patients, right? So we have a good first strike. We know how to reduce that tumor cell population. We know how to get patients into the state of NED, no evidence of disease. We're pretty good at getting to this point. Now, the MVP is called the minimal viable population, meaning what can we do to get this remaining tumor cell population in patients who have their disease recur or come back? What can we do to make that extinct, right? And so I kind of came up with, and what I'm working on currently is kind of a way of creating a second strike, or thinking about how to find a drug or a way to target that resistant cell population. And so kind of what we're working on, still kind of in the planning stages, but what I think would be an interesting concept would be to give our 10 weeks of MAP chemotherapy like we usually do. But I think we need to refocus our efforts on doing our genomic sequencing, our molecular profiling on this primary resection sample. Oftentimes when patients are diagnosed, we'll sequence the biopsy, but we often do not either repeat or try to sequence the resection for a number of reasons. It is a more difficult sample to do sequencing on because a lot of the tissue may be dead or necrotic. But for patients who don't have a good response to chemotherapy, we know about percent necrosis, like you're going to have viable tumor there that we can sequence. And one of the things that's being done a lot at many institutions now is we make PDX models, which are patient-derived xenografts, where we take a patient's tumor and put it in a mouse with the hopes that, well, all right, this can be like an avatar for a patient where we can create multiple mice with that tumor and try different therapies and see what works. So what I propose is, well, let's do sequencing of this primary resection, which is more reflective of the resistance or the metastatic potential of a tumor. Figure out if there are any amplifications, dependencies, anything we can target using genomic sequencing. Trial that out in a mouse, and we've shown that we can target some of these amplifications in mice with some work done by Alejandro Suicodero in his lab from a few years ago. And if we can identify something, show that it helps in the mouse from the patient's tumor, we can then offer that to a patient, perhaps as a second strike, as what we would maybe consider to be a maintenance therapy or a treatment that we would add on towards the end of MAP chemotherapy. So we're beginning to try to figure out how we can do this. But I think it's how we need to start thinking about moving the needle with osteosarcoma, because although this is I'll caveat it, it's 8 patients, but the pattern we saw it in almost all of the patients where there would be this kind of emergence of that resistant copy number clone. So maybe if we can target that copy number clone, then we can deliver a successful second strike and drive the tumor population into extinction and prevent relapse from occurring. I think I went a little over time, but with that, these are all the folks that have helped me along the way, and these are all the foundations that have helped support my work. I put my email here at the bottom, Twitter handle. Happy to answer questions, or if you want to reach out, email, Twitter, whatever it may be. Love to talk about this work and any ideas you may have. Thank you all for your time.
Thanks so much, Dr. Kinnaman. And we have a few minutes left, so if anyone has any questions, please just put them in the Q&A or chat, and we can make sure we get those to Dr. Kinnaman. And while we're waiting for anyone to add some questions, and I did, as you were talking, Dr. Kinnaman, I was trying to wrap my head around this whole idea of necrosis and chemosensitivity and how that kind of plays with this, because I think this is changing, but I think even, I know 4 or 5 years ago, kind of the common kind of understanding was like, oh, okay, great. If you have high necrosis, like less likely to relapse, low necrosis. But I think we've seen that that's not really the case, and that there are other biomarkers that are probably a better indicator of of relapse. So I'm just trying to wrap my head around this idea that you might have cancer cells that are not responsive to chemo and you have lower necrosis, but those cancer cells, maybe they don't pick up those characteristics that allow them to survive, and so there's no relapse. Whereas you might have cells that are very sensitive to chemo and high necrosis, but those potentially are likely to get through that bottleneck and proliferate. Is that, I don't know. I'm just trying to kind of.
I think so. I think what you're saying is, how do you square with, what you're saying with the patients who seem to have, that extinction event almost, right?
Yeah.
That really high necrosis, but still there are patients who have high necrosis and it comes back.
Yeah, and the flip side too, where low necrosis and no relapse.
Yeah, and unfortunately, I wish I had a better answer for you. I actually think those are the most informative and interesting cases to learn from, as to like, okay, like this didn't go as we typically see. Why is that? And I think that's like kind of one of the next, areas of, I think we're going to get those answers soon as the genomic data kind of begins to mature, because like we now know we have sequencing data for patients from 10 years ago almost, and now we can begin to kind of figure out what mutations, what alterations lead to better or worse outcomes. And it's kind of a very like active area in the community of like, what are the prognostic kind of risk factors? Because I think that's what it comes down to. Like, even if you have that 100%, 99% necrosis, there may be one cell that has a genomic feature that's just, chemo's not going to get rid of, and that's that. But, practically speaking, kind of getting back to my last slide, if you still accept the fact that in most cases, like less than 90% necrosis is not as good as greater than 90% necrosis on a kind of population level, those less than 90% necrosis will have more viable tumor cells as a result to profile and to sequence, and you'll have a higher chance of identifying or sequencing, because the sequencing success rate can be low with those samples, just because there's a lot of necrotic tissue. So the poor responders will have a higher chance of having good profiling and being able to put implant the PDX and those sorts of things.
Great. So big takeaway from this too was just for patients out there is getting that sequencing done, not just at biopsy, but also at resection, and then probably for every subsequent event, right, or relapse.
100%. That is, yeah, that's that. If you leave with one take-home message, that would be it. Like, talk to your treating doctors, like, hey, like, is there any chance we can try to profile the resection sample? I think that's a very reasonable ask, and could be much more informative than the diagnostic biopsy. And then yes, with any relapse or recurrence, like having that sent for profiling is extremely helpful.
And to your point though, about how depending on where you pull the sample from, you might not capture the full, any thoughts on how to get around that?
So I have ideas. I don't think there's a practical take home, unfortunately, for parents or families. What they do with some cancers, some adult cancers, is they'll actually take a little bit of DNA from different parts of the tumor and just combine it into one sample. Essentially, it's called like a master sample, a global sample, and then you do the sequencing of that, and that way you at least capture like all regions, and you could potentially characterize all the different mutations that are present.
Got it. All right. Well, we are at time. That was so informative and interesting. Dr. Kinnaman, thank you so much for joining us on Osteobites today and for making it better for pediatric cancer patients everywhere. More information on this and all Osteobites can be found on YouTube, on our website at mibagents.org, and at your favorite podcast place. Next week is the third Thursday of the month. So instead of Osteobites, we are going to be dropping another episode of Osteo, where Mia, yay, woo, and other junior advisory board members Camille and Vicky spill the tea on dealing with osteosarcoma from an AYA perspective. And next week, you guys are going to be talking about coping with humor and going to be reviewing different cancer memes, which sounds like it's going to be a very entertaining episode. So definitely check it out on our website. And same thing on our YouTube channel, on your favorite podcast app. And we are going to return with OsteoBites on Thursday, October 27th. We're going to be joined by medical oncologist Dr. Ben Nassib, also from MSKCC. And he's going to be giving updates on the clinical sequencing of 2138 bone and soft tissue sarcoma. So looking forward to that. You can find the rest of our OsteoBites lineup for 2022 and even going into early 2023 on our website. And if you have any ideas for future topics that you'd like to hear about, just share them with us at events@mibagents.org. Thanks again, Dr. Kinnaman, for spending an hour with us today. And Mia as well, always love having you. And to our sponsor, the Osteosarcoma Institute. And thank you for joining us on OsteoBites today. Check out Osteo next week, and we hope to see you on the 27th when we talk to Dr. Nassib. Thanks so much, everyone.